scar growth. So my motivation for choosing Maine Med was that I've always been really interested in science, and I'm thinking about taking more of a research track instead of going to med school, so I wanted to see what that was all about. I've always wanted a career where I can help people, and I chose this specific lab because I think that cancer research is really interesting. And also this lab, I'll talk more about this later, but they really do a great job of promoting women in STEM, so that was a really cool reason to why I chose this. And then my familiarity, I've never been in a real lab. I took AP Bio, but that was a lot of content and reading and not a lot of doing. So I was in the Reagan lab, and that's Dr. Reagan. She's awesome. She's the head of the lab, so there's different wings at MMCRI, and so her wing studies multiple myeloma. They focus just on that. So just, okay. So this got my official badge. This was our lab, and so this lab was really dominated with females in leadership, which was a really cool experience and a really cool environment, especially because you don't see this in a lot of labs. Most of the other labs are primarily male dominated, so that was a really cool environment. And then this is just a picture of part of the lab that I was in for 2 weeks. So just really quick, multiple myeloma is a blood cancer that grows in the bone marrow. This is just the thought I would explain it because I didn't know what multiple myeloma was going into this, so that was definitely a challenge. Was I kind of jumped on a moving train and had to figure out all of this jargon. My brain was just on for 6 hours every day because you have to keep up with what they're saying and try to understand what they're talking about. So, multiple myeloma is a blood cancer that goes in the bone marrow, and right now there's no cure, and is resistant to all current therapies. And so, the Reagan lab focuses on adipocytes, which are fat cells, and osteoblast lineage cells, which are just the cells that line your bones. And so, their research contributes to new therapies for the tumor growth. So that was really cool. And so originally I thought that I would just be kind of getting them coffee or reading papers while they did their work, but they put me in the schedule. Each day I worked with a different person on their experiments and on the different ways that they have been contributing to this this issue. So it was really cool because I got to directly do stuff. So this is just, you can't really see that well, but that's just normal bone marrow and then multiple myeloma. So this is it okay if I turn off this light?
Yeah.
Oh, okay. Should have wrote that darker, but it says scaffolds for tissue culture. So the Reagan lab is really unique in that Dr. Reagan invented this way. She raises her own silkworms and harvests the silk cocoons to make a protein scaffold, which is basically just like a model of the bone structure. And so it makes a 3D bone-like environment where they can culture the tumor cells and actually study the relationship between fat cells and the tumors. So that was my job. So they're constantly making these scaffolds. So this was the protocol that I received. And these are just a few steps of the process. One thing that I learned was that experiments take a long time. This was over the course of all the first week. You had to boil and rinse every few hours and just really take care of it. And so it was really cool because then after I made this, my job was to help. I had to train other people that were new to the lab the following week how to make this. So that was really cool that I got my own responsibility. So then another thing that we did was make cDNA, which is copy DNA. So I was helping this researcher study the effects of multiple myeloma on the fat cells and how that affected gene expression. So we took the DNA, which makes RNA, and then we made copy DNA of it, and then we put in a qPCR machine, which basically just heats up the cDNA, which is copy DNA, to high temperatures and then really low temperatures for hours, and it makes a ton of copies of it. It amplifies it, and then it was really cool that I was there, that we got to study the gene expression from that, and we found out that multiple myeloma reduces the gene expression of the fat cells. So that was cool that I was there for that discovery. And so we had to extract that tiny RNA off the top of that. So it's really scary. I learned to not mess anything up and using the different pipettes and things like that. It was a lot of pressure, but it was really cool to learn about that. Another thing that we did was take enzymes from fireflies and stain the human bone marrow stem cells, and that's the microscope room. And so these are the really beautiful images that we came up with from that. And so that was really cool to be able to take images of the actual stem cells that we were culturing. This is me culturing cells. It takes a while, something that I learned, as I said, with the scaffolding, everything takes a while. So day after day, we'd have to change the media, which is basically the nutrients that the cells grow in these hoods. And we'd be in there for hours just using the pipettes and kind of cleaning the cells, counting them under the microscope, incubating them. And then the last thing I did was we took mouse toes and genotyped them. So basically what that is just making a gel, and then you use this enzyme to disintegrate the mouse toes into just their DNA, and then mark those with some fluorescent markers, and then put them in the gel, and then attach electrodes to that gel, and then you can watch the things with less base, the DNA with less base pairs will go farther. And so what we came up with was this image, and it's kind of, you can't really see it, but there's little tags up there, and then there's these tags too. And so basically we were looking for mice that were heterozygous to mate and keep this mutant phenotype going in the mice litters. And so this is just a picture of the multiple myeloma cells exposed to a drug, but that's just an example of the cells that I had to count, which is a lot of cells to count. So that was definitely a challenge, getting used to counting all those cells and not messing up. Okay, so takeaway. I learned so much more than I thought I would. I really had I was very shocked when I went there because they kind of just put me right in. And so I had a lot more experience than I anticipated, but it was also incredible. I realized that I was able to develop lab skills and familiarity with the things in the lab before college. So that was really helpful because I learned that this is definitely something that I want to do, and I'm not sure that I definitely would be as comfortable in a lab in college. And so another thing that I learned is that research, I've heard, is really kind of lonely and like isolated, but this lab was incredible in that everybody worked together and everybody was so nice and cared and just supported each other, which I know not all labs are like, so that was a really cool environment to be in. I also learned about ethics and sacrificing mice to study myeloma tumors, and so that was definitely something that everybody agreed that although it is difficult to sacrifice mice, you're definitely helping in the long scheme of things to help find cures for this. And then I definitely want to pursue research, and definitely passion is key because you can dedicate years of your life to researching something and not really come up with a solution, but anything that you do helps other researchers, and you're always contributing to the field, so even if it's frustrating or takes a while, I learned that you just have to, if you're passionate about it, then it's all worth it in the end. Okay, sorry, I know I had a lot of slides, so I went through that kind of fast, but yeah.
Any questions for Christie?
Have you done anything with Maine Medical before this?
No.
Is it hard to find an STP like this? Like, how do you even start?
I just emailed. I just went on their website and emailed a bunch of people, and Dr. Aiken got right back to me. It was really cool. Yeah, it was super easy.